From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. This episode was recorded on August 1st, 2023. Hello, Micronauts, and welcome to our Quality Quorum. Today is the first episode of Matters Microbial, and we're all very excited. For our initial segment, I'll introduce myself and my own microbiological history, then talk a bit about my goals for the podcast. After that, I will discuss three important questions about microbiology and microbiologists with my first guest, Dr. Vincent Racaniello of Columbia University, Earth's virology professor and the podfather of Microbe TV. Folks, I thought I would begin this whole podcast situation with a little bit of my microbiological journey. I was born quite a while ago, actually, in Compton, California, to a lower middle class family. No one in my family knew anything about academia. My older brother and I were, in fact, first generation college students. But I was very fortunate because when and where I grew up in California, I was identified as someone who could benefit from enrichment. That's a microbiological joke. And so as a result, I had many opportunities that other people didn't have. And it didn't cost my family anything. And that was a wonderful thing. I did science projects throughout elementary school, middle school, and high school. I was fortunate enough, in fact, to go to UCLA as an undergraduate. And there, I loved biology. I loved everything about it. But the beginning of my junior year, I took my first microbiology course, and then it hit me how much I enjoyed the small masters, Matters Microbial. And my introductory microbiology professor was Dr. Sid Rittenberg. And Sid was one of the folks who was an authority, and this is kind of an interesting coincidence, in predatory bacteria, specifically Vibrio. And I was entranced during every single lecture. I learned so much. And I have to say that when I was fortunate enough to receive a Karski Award from the American Society of Microbiology a few years ago, on the list of Prider awardees was Dr. Rittenberg. So the circle closed. Following my undergraduate degree, I went on to Stanford University for my PhD. And I'd always been interested in symbiosis. And I was, of course, interested in microbes. In my opinion, one of the better ways to study symbioses. And turns out that a particular professor came to work at Stanford named Sharon Long. And I was her first graduate student. Now, what Sharon and her coworkers studied was the association between the bacterium found in the soil, Rhizobium, and the roots of leguminous plants. Now, what happens is those bacteria cause the plant to form a knot, a gall, something called a root nodule. And within that nodule, the bacteria, in a beautiful ballet of symbiosis, are able to take atmospheric dinitrogen and turn it into biologically available ammonia. The time that I began, not all that much was known about the nature of the symbiosis. So my basic work as a graduate student was to work with bacterial viruses of Rhizobium. I wanted to develop a system to move genes around to be helpful in genetic analysis. And I started working with a bacteriophage called M3, which is still being used in the study of now called Cynorhizobium meliloti today. And this was a wonderful thing for me. Now, I do have to kind of predestine a little bit of what's going to go on. I have always been most interested in what I call undomesticated microbes. That is to say, microbes that are a little more challenging to study because they don't have adequate tools. And as a result of that, there were lots of exciting things that I would find out, but it would be very difficult to study them. After earning my PhD, I moved to San Diego and I worked at both UC San Diego and a place called the Augeron Institute. And in association with Michael Silverman, I was able to do some work on Vibrio harveyi, a bioluminescent bacterium. And that was really wonderful to me. I can't tell you how much I enjoyed it. It was so wonderful to think about the regulatory genes that make light being produced, why that light was being produced, and why it's a beautiful blue-green color. To answer your question, it is the wavelength through which light transmits best underwater. So it kind of makes sense. In any event, at the conclusion of that postdoc, I didn't want to leave San Diego. And I was able to get a job in what I'm going to call a biotech company. It was called Kelco, later acquired by Merck. And what Kelco did is it took bacterial polysaccharides, and they were able to use them in both food and industrial concerns. And actually, you know the names of two of them. Xanthomonas campestris actually makes a polymer called xanthan gum that you can find in salad dressings. Another organism, then called Pseudomonas elodie, which I believe is Pseudo-Ultramonas elodie today, makes gellan gum that you can find in Snackwell cakes. This was not a very exciting situation for me because I really like working at the bench. I really like doing experiments. And in fact, when you work in an industry, you're more goal-directed, and I understand that. So it really wasn't appealing to me too much. So fortunately, I had made a lot of friends in the San Diego area who were science fiction authors because, unsurprisingly, I really enjoy science fiction. And one of those authors, Dr. Gregory Benford, became a very close friend of mine. And he suggested that we write a couple of novels together, which we did. I want to be very clear, there was not a lot of money to be had, but I did make a little bit of money with it. And this became very handy when, in fact, I was laid off from Kelco. And things got a little bit tough. And here I have to name drop. I had a friend in graduate school named Daniel Kleonsky, and he's a very well-known professor who studies apoptosis at the University of Michigan. And we'd stayed in touch all these years. And he wrote to me in these days before email that he had heard about a job that opened up to teach undergraduate students at a place called Occidental College in Los Angeles. So with only about 3 weeks of money left, I was able to get a job actually teaching students. And I taught Introduction to Cell and Molecular Biology. I taught microbiology. And I cannot express to you how transformative that was to me. I was able to take the microbial diversity course at Woods Hole. I was able to work closely with students. Please remember, listeners, that when I was at UCLA, it was a very, very, very large campus with a large population. In fact, my friend Dan Kleonsky had also gone to UCLA, also gotten a degree in biology at the same time I did, and we had never seen one another. That's not the case at the small liberal arts college. So I was really excited at every aspect of working with the students. And then after about, I should say that was the place I met and married my wife, had my 2 children, and really decided this was the career for me. And then after about 7 or 8 years of that, we moved here to Tacoma, Washington, where I currently have tenure in the Department of Biology at the University of Puget Sound. And it's a lovely place to raise children, a lovely place to work with students, a lovely place to do research. It's a wonderful place. I really like how green it is because if you know anything about me, I like tardigrades. There's a lot of them in moss here, so that's a big deal to me. I do want to point out to listeners that working at a small liberal arts college is a little different than you might expect. I don't have graduate students. I don't have postdocs. I don't have master's students. I don't have a technician. So as a result, I pretty much work with undergrads who have limited amounts of time. Now, this certainly shortens the amount of, or decreases the amount of publications I can get out, but it does mean that I can study whatever I want, and I can really bank on the enthusiasm of students. And I've been very fortunate because I call students who've spent more than 1 year working in my laboratory, A former student of mine, Kim Dill McFarland, called us Doc Martians, which is kind of funny, I guess. So I have sent 25 Doc Martians to PhD programs, 5 of whom have faculty physicians to this day, one of whom earned tenure at Stanford a few years ago. And I want to be very clear, I'm just the mentor. These are fabulous students who I got to know one-on-one and work with. So what I really am is I'm a talent scout, and I'm a good one. So that's my career. That's what I've been doing all these years. But I have always loved science, and I've always loved microbiology. And what really moved me toward microbiology at the age of 10 is the following. My older brother had a friend with the terrible disease of muscular dystrophy. And that friend's father was a carpenter and built my brother's friend a home lab. And I got to be his hands. And so we did pretty much anything we could out of Scientific American's The Amateur Scientist. And it was really a lot of fun and scratched every scientific itch that I had, all across all of science, ranging from astronomy to zoology. It was wonderful to work with Terry. And then one day, Terry gave me a Petri dish. And he says, take that into a dark room. And I did. And I saw that beautiful blue-green light. And I'll never forget that moment. And I think that's the bolt out of the blue-green light that made me into a microbiologist there. I should say again that all of my career, I've been most interested in undomesticated microbes. So currently, I do some work with predatory bacteria. Della Vibrio, taking us back to Sid Rittenberg, is one example. Also, encephalid herons. I also have maintained my interest in symbioses. I'm a co-PI on a grant with a colleague of mine in Tacoma, Stacy Weiss, where we study the microbiome that's associated with certain lizards. The idea being when lizards lay eggs, antifungal microbes are being deposited on those eggs to help protect them. This, by the way, is the closest I've ever been to doing something in research that might help people. I also do a certain degree of collaboration with another member of my department, Joel Elliott, who's interested in sulfur oxidizing bacteria that we find here in Puget Sound. And I am always on the lookout for those kinds of thiotropic symbioses. But it does take me back to bioluminescence. And as I've said, the last 5 years or so ago, I've done quite a bit of good work with bioluminescence with several of my students, and I've really enjoyed it. But I came across it in a different way. For many years, I've been interested in the intersection between science and art. And in the show notes, you'll see some images and some videos of the kinds of things that have taken place. And one thing that I learned to do years and years ago was to use luminous bacteria like paint and be able to create what I call lux art, which the ASM, likes to call auger art, that kind of thing. And it's a fascinating subject. The bacterium that I use, it's a species of photobacterium, was discovered by Eric Stabb and Ned Ruby in Hawaii. And it is so really wonderfully bright. And you'll see some of the photographs and some of the videos where I put on Lux Art competitions and Lux Art sessions. And I'm always struck by the amount of creativity that students have. And this is just the tip of the iceberg. I have found over and over again that involving students with creativity about science really sticks. For example, in my introductory biology class, I had a student who had terrible trouble with central metabolism, glycolysis, Krebs cycle, electron transport, and oxidative phosphorylation. So she created the game of life. Only it's about central metabolism. So she had game pieces and she did all this research on it. I have the board to that game actually put up on my wall, along with a lot of student art. And I'm here to tell you what a difference it's made to them. And I actually wrote a short paper about this that I'll put up in the show notes as well. So I guess what it comes down to is I really want to work with microbial literacy and really want to work with outreach, which is one reason I'm doing this podcast. I have an unfortunate excess of enthusiasm, but I promise you it comes from a good place. In any event, the goals for this podcast, as I mentioned before, are quite simple. I don't wish to take too much of your time. I'm appreciative of you listening. So there'll be about 10 or 15 minutes about a basic concept in microbiology or how it relates to a teaching experience. And then 15 minutes for an interview with a microbiologist and 3 big questions. The first is, what single event or experience turned you into a microbiologist? And you know my answer. What kind of microbiology do you, my guest, study? And why does it excite you? And thirdly, what is one thing that you would like the public to know about microbiology and about your research? So again, I do hope to have lots of fun things in the show notes for this. And now I would like to move on to my first guest. And now for the second segment of this podcast. It is my pleasure to introduce someone I've known for a number of years, Dr. Vincent Racaniello of Columbia University. Hi, Vincent.
Hey, Mark. Great to be here. I'm really honored to be your first guest on this podcast. Wonderful.
Numero uno, sir.
I just have a question for you. How do you look for tardigrades in the marshes, as you said?
Mosses is what I
Mosses.
Yeah. It's actually very interesting, Vincent. What you do is you take some moss and you put it in distilled water overnight, and then you bring up the water just with a simple hand lens, you can identify them.
Cool. That's great.
You probably read there was a recent report of a new species of tardigrade that was discovered in Japan in a parking lot.
Are they everywhere?
They're pretty much everywhere. I am pretty sure you'll find them in Antarctica, but I don't know anyone who's seen that yet. But I could be wrong.
Are they in the built environment?
Yes. My building is covered with mosses. So the fact that they can invade, and I don't want to worry people, but I think if we start to look at HVAC systems, I'm pretty sure they're working their way in there.
Could I find some here in my studio, you think?
Oh, I bet you could. And you know what? I've got some DNA primers where we could do some swabs and look for their evidence.
It'd be great. I'd love to.
Yeah, we'll have to do that.
Okay.
Hey, Vincent, would you introduce yourself to the listeners?
I would love to. I am Vincent Racaniello. I have been a professor at Columbia University for over 40 years. I started there in 1982, and I did research on viruses for many years. And last year, I closed my laboratory, and now I do full-time teaching and podcasting. And I am actually in our studio. We call it the Incubator in New York City. I come here every day, except when I'm teaching, and make podcasts. And this is what I'd like to do for as many years as I have.
I think the listeners should know, and you probably do know already, that Vincent has been such a powerful force for microbial literacy with This Week in Virology. In fact, well, I should say that's been very important as we negotiate the whole pandemic with COVID-19 and has been very useful to so many different people. But I would also say there are so many other types of This Week In that you do, so much so that I've been calling it Twix, with X being the other group.
That's right. I think we have 7 or 8 podcasts. I should really know. It's like not knowing how many kids you have, right? Not a good thing. So we have This Week in Virology, as you said. That's the first one I started in 2008, and which passed 1,000 episodes back in April. Then we have This Week in Parasitism. We have This Week in Microbiology, which Mark has been a guest on many times, and he is actually TWIM-centric because he's been on so many times. We have This Week in Evolution. We have This Week in Neuroscience. We have Immune. We have Infectious Disease, Puscast. We have Beyond the Noise. And all of those can be found on the YouTube channel. I also put my virology lectures there, and from time to time, individual videos explaining things. And we are in the process this summer of developing a couple more shows. So stay tuned for something a little bit different. All of our programs are rather long, and I would like to make some short content. So stay tuned for that.
I'll do my part with that, Vincent. So I'd like to hit you up with those 3 questions if I could, sir.
My pleasure.
All right. The first is, what is the single event or experience, one, that turned you into a microbiologist?
I was at Cornell from 1970 to 74, long time ago. I was a biology major because I loved biology in high school. I had a wonderful high school biology teacher. But my father wanted me to become a physician. He was a surgeon, actually, and I couldn't say no. So I ended up going through college. I didn't get into medical school. I think I only applied to one, and I wasn't interested. So I ended up graduating without anything to do. I had no plans. I just went back home, actually got a job in a microbiology laboratory, isolating contaminants and identifying them. And that was very interesting, but I could tell it wasn't going to be a lifetime thing. But during the time I was working in that lab, I read a book called Fever by John Fuller, which is about the emergence of Lassa virus in Nigeria in the 1960s, the first emerging virus that we learned of. And I was totally fascinated by viruses and by this idea that they could come from animals and infect people. I just didn't know that before. So I decided after reading that I wanted to be a virologist. But now here I am working at home. I don't have any advisors. I don't know what to do. So I applied to a couple of master's programs in the area. And one night I went to dinner in the next town at the house of one of my friends from Cornell. His name is Ed Kilborn, and his dad is also Ed Kilborn. He was chair of microbiology at Mount Sinai in New York City at the time. So after dinner, Dr. Kilborn Sr. came out and said, "So, Vincent, what are you going to do with your life?" And I said, "I want to be a virologist, but I have no idea how to do that." So he said to me, "Well, it's easy. You could just come to Mount Sinai and get a PhD. Forget the master's. Just get a PhD. So I said, okay. So this was August. The next week I went for an interview. Of course, it's already very late in the season. I went for an interview. I got accepted immediately. I had good grades at Cornell. And then the next week I started in the PhD program. And that's what got me on my way. And then the moral of that is you have to find mentors. So Dr. Kilborn acted as a mentor for me. And I listened to him. So you need to identify people who are going to guide you, but you have to listen to them. So you have to pick the right person. It's not so easy. I had heard about Dr. Kilborn for years from his son at Cornell. And I knew he was a giant in his field, which happened to be influenza viruses. So I knew I could listen to him. So that's the key. You have to find mentors throughout your career. And there are many, many more I had in subsequent years. And they're still my mentors. Unfortunately, Dr. Kilborn passed away a number of years ago, but he's probably still mentoring me in some way. But you find them, you listen to them, and you keep listening for your whole life. And that's why I am doing what I'm doing today.
That is a really lovely story. And it really does show the influence a person can have. It doesn't take much time to get you down a particular road. And what I've found in my years of education is that we all get so busy, it's hard to listen. And so I really think that what you suggested here is important, Vincent.
Yeah, it's a good thing that you just said, Mark. We get so busy, we don't listen. It's so true. I'm guilty of that myself. I have a lot of things on my mind and someone's talking to me and I'm not listening. But if you do listen, you'll never be bored. I have this top from a Snapple bottle, which is about 20 years old. It says, "If anyone is bored, they're not paying attention, which means
Yes.
listening. Because if you just pay attention and listen, you'll never be bored.
It's very true. I mean, I've received a certain degree of criticism over the years for being interested in everything, but it's exciting. And I find that if I ask people questions, I find out interesting things. Some people only ask questions to wait for their turn to talk, which is not the same thing.
I think the best thing about a question is waiting for the answer. Give them a lot of time to talk. And that's the skill of being a podcaster, in my experience, that you be quiet, actually. And if you have a guest, let the guest talk. We don't want to hear you talk. We want to hear the guest.
It's really funny. I've run several times among educators who don't like the expression, I don't know. So they never want to tell a student that. And I've never had a problem with that, because my not knowing something is an opportunity to learn something I didn't know.
I totally agree with that. That's great.
And we're not fooling anybody. I'll say to students, have you ever BS'd a professor? And they'll say, oh, yeah. And I'll say, has a professor ever BS'd you? And they'll sheepishly say, yeah. I said, so we're not fooling one another. If you look for things I don't know, and I say this when I teach, you'll find things I don't know, but you'll also see that I'm excited about it. That should be your goal. And that's genuinely how I feel. This is really who I am. And
excitement makes you a good teacher.
I think so. So even though you've already, I think, answered some of this, Vincent, the second question I like to ask is what type of microbiology did you or do you study, and why did it excite you? And I think we knew some of it already based on the book that really influenced you. But please.
Yeah, so as I told you, I got interested in viruses from the book Fever, which, you know, like anything, it's an entree into something, and then you learn about it. And if you're not going to be excited fully, you would not do it. But the more I got into virology, the more excited I got. So I got into a PhD program at Mount Sinai. I worked in the laboratory of Peter Palese, a virologist working on influenza viruses, and it was very interesting. It was a good time in virology. We had recombinant DNA methodology being developed. We could do all sorts of new things. In 1976, while I was in Peter's laboratory, we had the swine flu outbreak in the U.S., which was fascinating. Then in 1977, we had the H1N1 pandemic. The lab was a very exciting place with people visiting from all sorts of countries. At one point, we had me of Italian origin. We had a scientist from Japan, and we had a scientist from Germany. Just amazing. So I loved doing virology. My work went well. And then as a postdoc, I went to David Baltimore's lab to do more virology, because Peter said to me, okay, Vincent, you're ready to finish. What do you want to do? I said, I want to work on RNA viruses. They're the best. He said, well, then you need to go to the best person in the field. And of course, I said, who would that be? And he said, David Baltimore. He had just gotten a Nobel Prize with Temin and Delbecco. And David and Temin's contribution was to discover the enzyme reverse transcriptase. So a very exciting lab to be in. I was very fortunate to get in. And then I went and I said, what do you want me to do, David? He said, I want you to work on poliovirus. So that's what I did. I turned to polio for 3 years, did very lovely work, in my opinion. We had recombinant DNA. We had sequencing of genomes all being developed. So I cloned the poliovirus genome. I determined its sequence. I found that the DNA copy was infectious. And with those findings, I got myself a job and headed back to Columbia, where I spent 40 years working on poliovirus and other related viruses. Just to give you an example, shortly after I got to Columbia, one of my first students, Kathy Mendelson, identified the cell receptor for poliovirus. One of my next students, Rebao Ren, then made a transgenic mouse with that poliovirus receptor gene. It became a new model for human poliomyelitis. So we used those reagents over the years. Very exciting times in virology. And then last year, I decided I'd done enough. I'd taken up enough space. I started I got involved in writing a virology textbook in the early 2000s. I started blogging, and then I started podcasting in 2008. And I got more and more drawn into the concept of educating the world about microbes, because the internet gave us a good tool to do that. So right now, I would say I'm not an experimental virologist, but of course, always, once trained, you're always a virologist or a microbiologist. And so I draw upon my expertise to try and teach as many people as I can.
That's a really wonderful answer. And I especially like the enthusiasm that came across your face. And I want everyone to see that, because so many people in the public misunderstand science. And they don't understand that it's a profession like any other. And you talk to many people, and perhaps they don't love their job. I have yet to meet a scientist who doesn't love their job. Oh, they might not like the bureaucratic aspects of things, but finding out new things, seeing things that people haven't seen before, don't have to be important, but you got to see that. It's the same way when Leeuwenhoek first looked through a microscope and saw things that no one had seen before. How exciting.
For a brief period of time, it's only you that knows that.
That's correct. So it's pretty much, that's an answer to the third question in some ways too, Vincent. What would you like the public to know about microbiology and about your research?
And this is a point that was really hammered home by the pandemic. Many people did not realize that viruses are everywhere, and most of them do not make you sick. We are really influenced by the ones that do make us sick. Obviously, they can kill you. They can make you go into the hospital. They can make your life unpleasant. So most people, I heard, thought there were a few viruses that infect us and they make us sick, and that's it. But this idea that every living thing on the planet, including tardigrades, are infected with some sort of virus, and usually many different viruses, most of which don't harm them, it is unknown to most people. So that's what I would like people to understand, that viruses infect everything on the planet. Every living thing has multiple viruses. And we, for example, you and I, Mark, have at least a dozen viruses, probably many more in us, and we are fine. They are probably contributing to our well-being. Mark often will talk about the microbiome, all the bacteria that are in and around us. We also have a virome, and those viruses generally are involved in your well-being. Now and then we will get a virus from something else that makes you sick, but I look at those events as accidents. And yes, we've had a big accident for the last 3 years in the form of this pandemic, but the evolutionary trajectory of a virus is not to make you sick. It's to make more viruses. And so sickness is something that is a byproduct of the interaction. And so I think it's important to recognize viruses as part of the tree of life on the planet, and everything contributes.
You know, it's funny you'd say that, because I've made a joke, and Maggie Middleton, who you know of as Vexed Muddler, actually made me a cartoon of this, because I've been saying for some time the vast majority of microbes don't really care about you or I or people at all, not even as things to live on. I mean, from the upper atmosphere to the depths that as far down as we can drill, wherever there's liquid water and an energy differential, we find microbes of one form or another. So the ones that make us sick are like the juvenile delinquents of the microbial world. There aren't that many of them, but they get all the press.
That's right. Exactly right.
And it's really, especially in our social media time where things get trumpeted, and I'm no policymaker, I'm just giving my own opinion, but I think that's an issue. And then people get carried away with that. So to have context is really important.
So I think it's a good example to take the pandemic virus, SARS-CoV-2. This virus was existing quite happily in bats somewhere and accidentally got into people and found that people were a good host to reproduce in and spread from host to host, because that's what viruses do. And unfortunately, it made many people sick and killed them. But in 20 years, it probably won't do that. And it will treat us as it did its bat hosts before. It will be benign within us. So there is no advantage to making a host particularly sick for a virus. I would say maybe if you cough a lot, you could spread it better, but you don't even need to cough to spread viruses. Breathing will do the trick. So,
would you mind if I asked a follow-up question to that, Vincent?
Of course, never mind. Now, go ahead.
So I know that I teach that quite a bit of our very genome are kind of the evolutionary husks of viruses. And that's really true. I mean, even if you look at what's called the ALU sequence, about 300 bases long, that makes up 18% of our genome. And some people believe, and we'll find out if you agree with this, that these are kind of the evolutionary traces of old viral, not necessarily diseases, more like events. And I'm wondering about the back and forth between the virome outside and the virome inside. Do you have thoughts about that?
Well, about 11% of our genome is endogenous retroviruses. These are remnants of old infections of either Homo sapiens or some of our ancestors, where the viral genome integrated into our DNA and remains there. All of them are not infectious. They've all mutated over the years so that they're not infectious. But as you know, we have borrowed some of those genes for our own purposes. So the placental protein syncytin was borrowed from one of those retroviral genomes that integrated into us. A large part of the genome is also these elements that you said, ALU sequences, lines, long interspersed nuclear elements, short interspersed nuclear elements. These are elements that can jump around the genome and they amplify themselves as a consequence. I don't think those are remnants of viruses. I think those are elements that are driven by reverse transcription that move around the genome. And they may have been the precursors of retroviruses because there are some that are moving around by reverse transcription. There are other pieces of viral genetic material in our genome. They're usually small, broken fragments, and so their significance isn't really understood. I think in the bacterial world, there's much more evidence for viruses integrating and contributing to gene flow and so forth. But much less, I think it happens for eukaryotic organisms, but we're much less aware of it.
Well, it's just such a, I used to call these dorm room conversations where you talk about where things came from. But there's still so much fun to think about and speculate, especially as we start teasing out information that supports or disproves thoughts that we have. And that's why it's, go ahead.
I think the fun conversations come when you have different viewpoints, right? You can have me and you and some other people all contributing their own viewpoint, their own facts. That's when it gets really interesting. And that's why I like doing podcasts, because you get a bunch of people together and just talk and good things happen.
Well, Vincent, it has been really lovely. Do you have anything else that you'd like to say before we close this session?
I want to wish you luck with this series, Mark. I'm looking forward to seeing it develop. I'm going to listen to every episode. I'm proud to be producing it at the Microbe TV production house, if you will. And I wish you a lot of luck, but I don't think you need it because you're a great conversationalist, and I'm looking forward to all the guests you're going to have.
Oh, we're going to have a lot of fun. At least I hope so. Thanks again for your support.
You're welcome.
Bye, Vincent.
Bye-bye.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes for today's episode can be found at microbe.tv/mm. If you like our work, please consider contributing to us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Vincent Racaniello can be found at microbe.tv. Many, many thanks to Julia Russo for superb editing, and Reba Clark for the fun music. I hope you've enjoyed being part of our Quality Quorum today. See you next week on Matters Microbial.